oceans make up over 70% of Earth and is home to 240,000 species of ocean life, with more to be discovered. World Wildlife reports that 200 million people rely on freshwater fish as a source of food, and 60 million people rely on fish as a source of livelihood. But what happens when the source of food, income, and livelihood disappears? Populations of freshwater fish are collapsing, with almost 1/3 of all freshwater fish facing extinction, and CBS reporting that total extinction of saltwater fish occurs in 25 short years. Overfishing, water pollution, and microplastics are some of the many issues accelerating this extinction. Overfishing is the process of collecting fish at a faster rate than the fish population can replenish at, forcing populations to deplete. The Nature Conservancy estimates that 4.6 million fishing vessels occupy our Earth's waters, with disposed fishing gear killing vulnerable species. Discarded fishing gear has been dubbed ghost gear, as the nets, lines, and traps discarded in the ocean often harm marine life that would otherwise be safe from fishing. Another form of pollution caused by fishing boats is known as trawling. Trawling disrupts the homeostasis of our waters by kicking up microbes and removing sediment with key nutrients to aquatic life. These forms of water pollution can be linked back to the excessive amounts of fishing boats in our oceans. Microplastics also donate to the issue of harming aquatic life. Plastics have been designed as a malleable resource that can be functional even in small particles. When these small particles get into our oceans, marine life mistakes it as a food source, and the plastic ends up in their digestive system and littering waters they inhabit. As this plastic accumulates in marine habitats, protein and minerals in fish reduces, as reported by Dong-Fang Deng, a professor at University of Wisconsin-Milwaukee. The solution? Cellular agriculture. Cellular agriculture has been at the forefront of discussion recently, as scientists have discovered a way to artificially grow proteins in a lab. Specifically, the use of salmon cells has been leading the industry, which begs the question: with the implementation of new cellular technology, how might food become accessible to a greater population, and how might this new technology cut down on environmental pollution? The company Wildtype has attempted to answer this question through the creation of lab-grown salmon, devoid of microplastics, eliminating fish gear and ship pollution, combating the issue of overfishing by manufacturing the protein itself and planning to reduce cost so that this protein is more accessible to the general public. Although their method is yet to be revealed, a Nature article discloses their own practice of cultivating sea life cells in order to recreate edible lab-grown protein. In the Nature article, researchers have created a method of sustainable and harmless cell collection and growth. The method? Collecting cells from fish fins, a part of the fish that is commonly discarded, regenerative, and unharmful to the species and the ecosystem surrounding them. In order to understand the collection method, we must begin with an understanding of cell biology. Pictured here are normal fibroblast-like cells, skeletal muscle-like cells, neural-like cells, and neurofilaments. These cells were differentiated from DESC cells to create various types of cells that could be repurposed into protein. These images were captured with the use of immonofluorescence and scanning electron microscopy. The adherent cells used in scanning electron microscopy were fixed with 0.4 milliliters of 2.5% glutaraldehyde and were kept overnight at 4 degrees Celsius. The differentiated cells were placed in 4% paraformaldehyde for immunofluorescence. Fish have the ability to regenerate certain body parts, including fins. By clipping the fin of a fish, researchers are able to isolate cells and culture the cells into a piece of protein that not only tastes like fish, but researchers create a mold that appears like the sashimi you would expect from a sushi restaurant. Authors Yu Suka Sirawaka and Eriko Shimada took cells from thread sail filefish, with their scientific name known as Staphylolepis sirifer. Fibroblast cells were then derived and subsequently immortalized. These cells were then analyzed to ensure that they did not possess any chromosomal mutations, and named DESC cells, standing for dedifferentiated Staphylolepis sirifer. Next, these cells were differentiated into fibroblast, skeletal muscle, neural, and neurofilament cells. After selecting fibroblast cells as the foundation of the cultivated protein, scientists pursued an environment in which these cells could grow into a food source. They discovered that these cells were capable of neural differentiation, with a serum composed of KBM neural stem cell medium and neural induction supplement. After discovering that these cells were able to proliferate, scientists had a new dilemma: how to turn cell body cells into tissue that is edible. These cells were placed in a flask, and were discovered that they would be able to culture both adherently and in suspension. The DESC cells were then cultured and stacked into sheets known as adipocyte cell sheets. The sheets were then subjected to a physical test in which researchers poked with a spatula to detach cells. The sheet responded by curling its edges. From this, the researchers then created a mold to shape the cell sheets into an appealing sashimi-presenting shape. With the application of a simple sensory test, researchers found that the protein had no unpleasant features and could be a feasible new source of food. They recommended that any food source to be generated in the lab must meet the following criteria. 1. Ease of procurement of raw materials or ingredients. 2. Ease of processing. And 3. Sustainability. The goal of creating lab-developed food is a concept known as clean meat. Food that has been manufactured as clean meat eliminates pollution involved with fishing, population extinction, and microplastics within our food. For now, Wild Type is working to perfect its product and drive costs down to be more affordable and accessible to the general public. Food accessibility is a growing issue as the human population has grown exponentially towards the capping capacity. With this promising new research, scientists hope to produce food that is not only wholesome, but sustainable for our Earth and accessible to all people, closing the gap between financial disparity and food security.